please. Hello, good afternoon, everyone, and welcome back. So I would like to call everyone and to take a seat so that we can start this second section, where we will discuss the roles and the future directions for immunotherapy. So it is my pleasure to introduce the two chairs of the sessions. We have Mark Yarchon, who is an associate professor of medical oncology at the Sidney Kimmel Comprehensive Cancer Center at the Johns Hopkins in Baltimore. He runs an NCI-funded laboratory, and his research is focused on developing novel immunotherapy combination for hepatobiliary cancers. And he will be joined by Greg Lesinski, who is a professor and vice chair of basic research at the Emory University. He also serves as the Winship Cancer Institute's associate director for basic research and shared resources, and where he provides oversight and direction for development and growth of Winship's basic scientific activities across the Centers for Research programs. So I will leave the session to you. Thank you, guys.
All right, good afternoon, everyone. It's a pleasure to introduce the next section on immunotherapy, which I think all of us are aware has become really a pillar of cancer care and a pillar of cholangiocarcinoma cancer care. Our first speaker is V. Sahai, who's an associate professor of internal medicine and the section head of GI oncology at the University of Michigan in Ann Arbor, Michigan. And his session is a clinical introduction on cancer immunotherapy for cholangiocarcinoma.
Thanks, Mark. I thank the chairs and the CCF for the opportunity to talk. I thought when Mark emailed me, he was asking me for longer lectures. I prepared 45 slides. I only see 10 minutes here. Oh, I think they've just gone up, right? 20 minutes now. So I appreciate you bearing with me here. Thank you. These are my disclosures. All right, so I was given a very focused task, and when I think about targeting biliary cancer, these are the ways that come across. We've talked about a lot about chemotherapy, cellular signaling inhibition, certainly not about epigenetics and mitochondrial to the extent, although there was a lecture earlier. So my topic is to understand the immune microenvironment, and within specifically the immune microenvironment, the immune regulation. So this is a top-level view of how we should interpret and how we do interpret immunity in our body. There's innate immune response and there's adaptive immune response. And there are two activating and inhibitory pathways that help us use these two signaling pathways. And there are specific receptors and pathways within this that we've been able to identify that we can further target to our advantage. And there are many different immunotherapy strategies. So when somebody says immunotherapy, to me, that doesn't mean much. I think within immunotherapy, there's immune checkpoint inhibition, which is really listed in the bottom as antagonistic antibodies, the way we think about anti-PD-1, anti-PD-L1 antibodies, anti-CTLA-4, and so forth. But also things like chemotherapy or immunotherapy strategies too, or agonistic antibodies, and others that are listed here. And there was a topic, or at least a discussion earlier about adaptive T-cell therapy. So this is the normal homeostasis within our body, right? The antigen-presenting cells are bringing the PDL or the ligand to the receptor, and that turns the system off. Now, the immune microenvironment of the tumor is unfortunately smart, and the tumor cells co-opt some of these ligands to present to the receptors and turn off the immune system within the microenvironment. And what we've been able to do so far over the past decade or so is to introduce these anti-PD-1, PD-L1, and CTLA-4 antibodies to block this pathways so as to allow the immune system to be on. So take the pedal off the brake, so as to say. There have been many, using this thought process, there have been many studies in biliary cancer, initially as monotherapy. The Keynote-028 was the initial study. Response rate was about 13%. And of course, a follow-up study was the 158, and that showed, unfortunately, the response rate was as low as 5.8%. And a median progression-free survival for those folks was only about 2 months. And other studies have been done, especially Dr. Kim, who studied it in 34 patients and noted a better response rate, about 11%, and a median PFS of 3.5 months. Similarly, other studies that I'm not going to go into detail have been done, all with monotherapies across the world. And then eventually dual antibody studies with anti-PD-1L1 and CTLA-4 was done, and a response rate, unfortunately, was very poor still at 10-11%. So what about chemoimmunotherapy? What if we give chemotherapy that destroyed the tumoral cell, produce these neoantigens that the antigen-presenting cells will then take over and allow the immune system to better identify these tumor cells and kill those by release of certain chemicals? So using this strategy in multiple cancers, including in biliary cancer, there were many trials, and this was one of the first and only trial in the US with chemoimmunotherapy that we did several years ago. And there were 2 arms on this study, chemotherapy plus nivolumab and nivolumab plus ipilimumab. And it was a randomized trial, and was published in the Cancer Journal, and showed that at 2-year mark, we were able to, compared to historical data, double the number of people who were still alive. And using these data, as well as other data from other trials done across the world, there was many phase 3 trial discussions, and 2 of them eventually went forward. And the TOPAZ-1 data, which has been presented before, which I won't belabor, showed that there was an improvement in median overall survival for our folks in the arm that had the immunotherapy in combination with chemotherapy. And specifically, the 2-year overall survival rate was indeed doubled in our patients. In the subgroup analysis, which often comes up as a question, what about the tumors that actually present more anti-PD-L1, present more PD-L1 expression? And indeed, those folks did better, but it does not mean that if you don't have a high PD-L1 expression in your microenvironment that you will not respond. Those folks also responded. What about the region of the world? If you look at the forest plot here, folks in Asia apparently seem to do better compared to the rest of the world. And the group was somewhat similar, 52% versus 48%. So we quite don't understand why the Asian subset of patients did better. But again, the hazard ratio is still below 1. And even though the confidence intervals cross, the study was not powered to answer this question. It's just of importance to know this as we discuss with all our patients in clinic. And more recently, similar study design, with the exception of continuation of chemotherapy rather than stopping at the 6-month mark, was the Keynote-966, and it again showed in a larger patient subset, about 1,000 patients, that indeed these folks did better with the addition of anti-PD-1 antibody. But what about second-line immunotherapy? So we did this trial now. We're in the midst of writing it up, and presented last year at ASCO. And it was 5-fluorouracil, liposomal irinotecan, and nivolumab in second line for a patient population. And we showed similar data to the one that was earlier alluded to, the NIFTY data that was presented in, that was done in Korea, South Korea. And our data is all in the US, with a median progression-free interval of about 5.4 months and an overall survival of 8 months, which is at par with the NIFTY data. Although the combination is safe and well tolerated, it may be active in the US population. We really don't have a cohort to compare the chemotherapy in the US. Dr. Archon's data and Dr. Azad's data, wherein they took patients with unresectable cholangiocarcinoma and took a very novel approach based on preclinical data. What if we combined anti-PD-1, PD-L1 antibodies with a MEK inhibitor? And the data was quite intriguing. In fact, it led to a subsequent study, which is ongoing in the US, and I think is enrolling quite robustly. Other combinations have also been used and studied, although there's not really great activity in combination with anti-VEGFR2 monoclonal antibodies in biliary cancer, as was seen in HCC. Needless to say, there are many, many trials going on with immunotherapy in our patients, which is fantastic. Good to hear with several different combinations, and I won't belabor these tables much. What about predictive biomarkers in these patients? How do we put them into perspective? So this was data that was presented from the MSI high patients, microsatellite instability high, or deficient mismatch repair. And these are folks who had a response rate as high as 31%. So if you have an MSI high, if you have a deficient MMR, you need to get onto this therapy, currently studied as second and beyond, but may eventually make its way to first-line therapy as well. Within cholangiocarcinoma subset, there were of 22 patients. The response rate noted here was 41%. And I think the updated manuscript, which was just published a few weeks ago, showed a response rate was 51%. And 4 out of these 22 patients actually had complete response, no evidence of disease. And the survival was, median progression-free survival was not as robust as I would have expected, but certainly look at the tail in the curve. Majority of these folks actually did quite well for a long period of time. So the duration of response was quite intriguing. And the median overall survival is certainly laudable and something to look forward to for all our patients, not just this cohort. How about the high tumor mutational burden? So these are folks who have got a lot of mutations in their tumor. And how do we identify them? Through the same genomic analysis report. There is some overlap between MSI high as well as the high tumor mutational burden, but it's not more than 20%. Out of the 100 patients in this tumor mutational burden high group, about 20% had microsatellite instability, and the rest, 80%, did not. And these folks responded similar to the MSI high. And the last column on this is the non-high patients, and they did not, unfortunately, respond to the single-agent immunotherapy. And again, very small fonts here, but just look at the differences between high tumor mutational group and the low tumor mutational group, and you see absolutely these folks are responding to the therapy, and they should absolutely be considered in clinic for this therapy. What about some of the other biomarkers? And again, some of this data was presented earlier today. It makes my job a little bit easier here. That the TOPAZ-1 phase 3 trial biomarker subset looked at about 64% of patients on which tissue was available for them to do this kind of analysis. And generally, there was no difference between any biomarker cohort that could be clearly identified, albeit some of the numbers were quite small for some of these cohorts, specifically for the mutated ones, and it was very hard to clearly make a difference. But from the data we have, we were unable to clearly say that these patients with IDH1 or FGFR or BRAF should be preferentially getting immunotherapy. And again, no difference in overall survival or progression-free interval. What about mutant IDH1 that I just referred to? In 2018, there was a Nature paper that looked at the combination of IDH1 inhibitor with the anti-PD-1 antibody. And they showed that the mice who received the combination actually did better in terms of survival with a significant p-value as shown here. That led to the ivosidenib and the nivolumab trial, which is ongoing. And then more recently, Dr. Bardisi's work showed that actually it's the CTLA-4 checkpoint that is more important than the PD-1 checkpoint, and we should target that, which is, again, hopefully leading to, and I think is leading to this trial, which is listed here, ivosidenib plus nivolumab plus ipilimumab. So if you have an IDH1 mutation, please make it a point to get on this study. It's extremely important for you as well as for this study to move forward quickly so that we can apply these results to the rest of the world. How about, excuse me, the DNA damage repair pathway? There are many different mutations that fall within this, and I've included IDH1 and 2 in this group as well. But in addition, some of the common players in DNA damage repair pathway that are talked about are the BRCA1 and 2 mutations, ATM, and maybe MUTYH and BAP1, although the studies for the last two have so far not panned out. So when we looked at this in 2015, we found, and Dr. Tran, who's in the audience here, is now left for greener pastures to Mayo, did all of this work. And we found that maybe 28% of these patients would respond to PARP inhibitors. And so we hypothesized that what if PARP inhibitors in this patient population could cause DNA damage and lead to more neoantigen production? And if that is true, then maybe the combination with immunotherapy should be considered, because that would allow the immune system to better recognize these cancer antigens and therefore kill the cancer cells. So with that, we ended up designing this study as listed here, and we took patients with all patients with biliary tract cancer, wanting to identify post-hoc after the fact which patients did better, whether these were all the expected players as noted before, or some other patients also responded. And this is just a preliminary data that I wanted to show that 3 patients on that study had the best response. One of them had a partial response, but if you look at it, all 3 patients with an IDH1 mutation stayed on therapy for over 8 months. And that was not true for 2 of the other patients who had olaparib as monotherapy, and in the midst of writing this up. Can we leverage what we know today for a better clinical trial in these folks? And an example of this is listed here. We're calling this the MBEL trial. As you know, after 6 months of chemotherapy and immunotherapy, we are supposed to, as per the TOPAZ-1 trial, transition to durvalumab alone. But what if at that point in time, we started recognizing the mutations that are present? And if there's a HER2 high patient population, can we add trastuzumab to the durvalumab? And there's a lot of data to suggest and learn from breast cancer. How about the BRCA1 and 2 mutations? We just talked about it. PARP inhibitors cause DNA damage. Add them to the immunotherapy. Allow them better recognition. We just showed the data about IDH1. FGFR2, I'm not very convinced as of now. It's also cold, just like IDH1. It may or may not have efficacy. BRAF V600E mutations. So melanoma, the current therapy in melanoma, at least one of the current therapies in melanoma for first-line therapy is BRAF inhibitors plus MEK inhibitors plus immunotherapy. So why not do a maintenance targeted therapy trial? As part of other works, we've looked at other markers that could help us identify improved role for immunotherapies, more biomarkers. That's exactly what we need. So we identified that the BRAF patients actually have a very, very high CD73 expression. And we showed in many different ways across many different subsets and much higher than the KRAS population. So the study that we are proposing is combination of anti-CD73 antibody, which is in clinic right now, with durvalumab, with the primary endpoint of overall response rate. I haven't covered all the topics because of time, so I'm just listing here dietary fiber. So preclinical data, very strong data, that if you're on immunotherapy, you need to have a high fiber content in your diet. There's a trial going on. If you're interested, be part of that study. Otherwise, please take in high dietary fiber if you're on immunotherapy. We talked about the dual PD-1 HER2 blockade just a few minutes ago. This is shown to be positive in gastric cancer and previously in breast cancer. There's studies that are coming up with fecal microbiota transplant to overcome the resistance to anti-PD-1 therapy. It sounds terrible, but it works. And then very recently, the POL mutations, they may be another genomic biomarker for improved efficacy for checkpoint inhibition. So key takeaways and emerging questions that I want to point to is certainly the phase 3 chemotherapy plus immunotherapy trials have led to improvement in survival, doubling of survival rate at 2 years, and define the new standard of care for our patients. There are ongoing multi-omic characterizations that will allow for further treatment stratification for immunotherapy, and an example is the IDH1 mutated patient population, and we use the inhibitor plus the CTLA-4 antibody upcoming trial. And novel immunotherapy trials are critical. An example of that is adoptive T-cell therapy trials. Several of them are going on at the NCI. I think every other week I'm referring a patient for those trials. So please do consider them as part of your repertoire. So lastly, just wanted to point out one of my patients that I've treated for past 2 years was able to convince the governor of the state of Michigan to declare February as the awareness month. I had no idea that was not the case, but we finally have it recognized as a Cholangiocarcinoma awareness month. So kudos to you guys. Thank you.
All right, thank you for a wonderful presentation, Dr. Sahai. At this time, I'd like to introduce Dr. Lleo, who is a professor of internal medicine and the head of the residency program in internal medicine at the Department of Biomedical Sciences in Humanitas University. She has assumed the responsibility of the hepatobiliary immunopathology laboratory in Humanitas, and she'll be discussing high-dimensional profiling of immune cells in human cholangiocarcinoma, what to target.
Thank you, Greg, for the kind introduction. Thank you for the invitation, Chiara and Laura, and thank you, CCF, really, for hosting this amazing meeting. These are my disclosures. So I'll go very fast with the introduction. We know everything about cholangiocarcinoma. We know that despite being classified based on their anatomical location, it's a very heterogeneous tumor in the molecular presentation, in the mutations that are present. The important thing I wanted to point out is that, as we know, these mutations are associated to the prognosis of the patient. But what do we know about the actual immune microenvironment of cholangiocarcinoma? And I think this question is key now, in which we are facing, as we know, the immunotherapy era in cholangio. From now on, I will focus my presentation mostly on intrahepatic cholangio, because data on extrahepatic and gallbladder cancer are scarce, and also because the work we've done and that I will be presenting is focused on intrahepatic cholangio. So data from Daniela's here, dated 10 years, showed for the first time that there is a large subgroup of patients, and probably, yeah, I have a pointer, which are almost 40% of them that have a inflammatory pattern. These data were based on integrative genomic analysis of 150 patients. Importantly, these patients express high levels of genes that are associated to the immune response, and this subtype of cholangio is actually associated to a better prognosis. We did a very simple study some years ago showing that by immunohistochemistry, the infiltration with CD3 T cells, CD8 T cells is associated again with a better prognosis. Again, a novel immune microenvironment-based classification of cholangio showed, based on the infiltration of different cells from the immune system, that more hot tumors have a better prognosis. These data were also confirmed recently from a group at the MD Anderson, and again, the group of Daniela Sia from New York was able to further divide these subtypes of intrahepatic cholangio, showing that the most inflamed cancers have a better prognosis. So we know that within this heterogeneity of intrahepatic cholangiocarcinoma, the molecular classification is important because the type of immune infiltrate is associated with the prognosis. However, we lack integrative information regarding the interaction of the different immune subtypes within the tumor, and also the genomic background that drives different phenotypes of these cells. So in my lab, we decided to perform a study in which we first perform an extensive phenotype characterization of cells from 20 patients with intrahepatic cholangiocarcinoma. We isolated CD45 cells, which are the cells that compound the immune infiltrate, from the tumor, from the peritumoral, from the peripheral blood, and also from the what we call the blood perfused from the tumor, which can be identified as the intrahepatic circulation. So 4 different subtypes, we wanted to see exactly what was the composition of the immune microenvironment in these tumors. After that, we wanted to see the tumor-driven signatures of these different populations to see if there was a correlation with the clinical outcome. So starting from the first part of the study, as I told you, we have these 4 subtypes. We were able to see that these 4 tumor sites have different T cell and myeloid composition, that actually the more similar are the peritumoral and the perfusate, showing that the intrahepatic circulation is actually very similar to the tissue-resident cells. But if you look at the different cell types that are more abundant within the tumor, you can see that we have a lot of tissue-resident CD8 and CD4 cells, but a large amount of T-regulatory cells. If you look at specific, oh, sorry, I went a little bit ahead, but it's okay. If you look, again, focusing on the Tregs, since they were the more abundant T cell subtypes, we decided to focus on them. We were able also to see that there are different clusters of T cells. Specifically, we have quiescent and activated Tregs. All of them express FOXP3, so there are classical T-regulatory cells. Taken all together, this data confirm that there is still a poor immunogenicity of human intrahepatic cholangiocarcinoma, although there are some subtypes of cells that are more abundant compared to other types of tumors. There are still not many cells, and that the Tregs that infiltrate these tumors have a heterogeneous pattern with a phenotype that is similar to what has been previously described in other tumors. So we moved into the transcriptomic analysis of these cells. We wanted to see exactly what was the gene signature that was driving this differentiation in T regulatory cells and in these cell subtypes. As you can see, T cells and NK cells are more abundant in the tumor compared to the peritumor. But we were able to identify 10 different clusters of T cells, and again, confirming the data from our FACS analysis, the T regulatory cells are much more abundant within the tumor compared to the healthy tissue. So at this point, we looked into the pattern of gene expression, comparing all the clusters of T cells within the tumor and in the healthy tissue to see if there was a different expression of inhibitory molecules or effector molecules that actually were characterizing these cells. And as you can see, in all the different clusters of T cells, there are differences. Importantly, we highlighted here all the most important genes that were differentially expressed in T regulatory cells within the tumor and in the healthy tissue. This data suggests actually that there is a functional modulation of the T cells within the tumor. So we see the different clusters, but these clusters have expression of markers that are different within the tumor compared to the healthy tissue. This seems like a complicated slide. What we did look here is the interaction, the cell-cell interaction of the T regulatory cells with all the other cells. And this data show that the Tregs are characterized by an extensive remodeling of the expression of the ligands receptor pairs that are required for the cell-cell interaction, suggesting that there is an enhanced Treg-mediated immunosuppression within the tumor. So we have a lot of Tregs within the tumor, but these cells actually mediate an immunosuppressive microenvironment. So at this point, we saw that there are a lot of Tregs, that these Tregs are actually have a different pattern of expression, but what are the networks of transcription factors that regulate these Tregs? So we looked into that by bioinformatics analysis. We identified a number of transcription factors that were differentially regulated in T regulatory cells. And we decided to focus in on one of them. It's called MEOX1 for two reasons. One is because, as you can see here, the expression is almost exclusive of T regulatory cells, and is actually, it follows the expression of FOXP3, which is a very classical marker of T regulatory cells. This transcription factor has already demonstrated to be highly expressed in other types of tumors, and is involved in the regulation of a complex network that involves other transcription factors. But we wanted also to have proof of the involvement of this transcription factor in the activation of Tregs. So what we did is we used T cells from a healthy donor, stimulated them to develop a Treg phenotype, and then make a, sorry, I got distracted. Transfected them to have them express high levels of MEOX1 associated to a GFP marker. That GFP expression allowed us to then sort these Tregs, the ones that were expressing high levels of MEOX and the ones that had low levels of MEOX. And through an extensive characterization of transcriptomics and epigenomics of these cells, we were able to see that this transcription actually recreates the phenotype that we see within the tumor, showing that it's actually MEOX-driven and that this could be a transcription factor that hypothetically could be targeted. The question is then, does MEOX1 expression in Tregs actually has an influence in the progression of the disease? Do these T cells that have, T regulatory cells that have high levels of expression of MEOX actually have lead a worse prognosis in the patients? So to do that, since our cohort was small, we used public data from a large cohort that had been already published. And as you can see here in these curves, the expression of MEOX was associated to a worse prognosis, also in the multivariate analysis. So what we can see is that in intrahepatic cholangiocarcinoma is characterized by a poor immunogenicity, that it's highly infiltrated with T regulatory cells that have a highly immunosuppressive phenotype. This immunosuppressive phenotype is driven by MEOX1 expression that strongly correlates with the overall survival of intrahepatic cholangiocarcinoma. Obviously, we need still to better understand the complex transcriptional architecture of this intracellular crosstalk. It would be very nice also to have similar data from patients that are being treated or are enrolled in clinical trials with immunotherapy-based regimes in order to see exactly if the immunotherapy is able to restore the original microenvironment. And with this, I thank you all for your attention. I want to thank my group, our collaborators, our funding agencies, and the CCA Foundation for having us all here. Thank you.
All right, and I'd like to invite Sumera Ileas up to the panel. So Sumera Ileas is an assistant professor of immunology and medicine at the Mayo Clinic, and she directs the hepatobiliary neoplasia clinic there. And she runs an NIH-funded laboratory focused on the immunobiology of cholangiocarcinoma and on the development of combination immune strategies for cholangiocarcinoma. She'll be giving us a talk specifically focused on myeloid cells as a mechanism of resistance to immune checkpoint inhibitors in cholangiocarcinoma.
Thank you, Mark, for that introduction. Thank you, Chiara, Laura, Stacy, and the CCF for the opportunity to present this work. So we learned, So we just got an overview on the tumor immune microenvironment of cholangiocarcinoma, and this talk will focus very much on a subset of immune cells that we think are really important in terms of patient outcomes, in terms of response to immune checkpoint inhibition, and there's a lot of interest now in immunosuppressive myeloid cells. We know that a high monocyte lymphocyte ratio is a predictor of poor outcomes, poor patient outcomes. We know that cholangiocarcinomas are very dense desmoplastic tumors that have an immunosuppressive milieu, which is characterized by increased expression of immune checkpoint molecules such as PD-L1 on tumor-infiltrating immune cells. And in this immunosuppressive microenvironment, there is a preponderance of immunosuppressive myeloid cells, such as tumor-associated macrophages, or TAMs, and myeloid-derived suppressor cells, or MDSCs. So this talk will really focus on these two types of myeloid cells. And we also know that response to immune checkpoint inhibition monotherapy, we saw the data earlier with Keynote 158, in CCA has been disappointing, and there's some data to suggest that these immunosuppressive myeloid cell types play a role in that. In terms of the tumor immune microenvironment of cholangiocarcinoma, so this is a very nice figure that was published in a review led by Tim Gretton, and on the left-hand side of your screen, you can see that the cell types that are anti-tumor, these are cytotoxic cell types such as CD8 T cells, tertiary lymphoid structures. And then there are a lot of immunosuppressive cell types, such as tumor-associated macrophages, MDSCs, as I mentioned, but also FOXP3 regulatory cells that we just learned about, as well as tumor-associated neutrophils, or TANs. Tumor-associated macrophages, or TAMs, are the predominant immunosuppressive cell type in cholangiocarcinoma. TAMs play a very important role in cancer initiation, progression, invasion, migration, metastases, and angiogenesis. And in the context of cholangiocarcinoma, TAM infiltration is associated with poor prognosis and chemotherapy resistance. And Stuart Forbes and colleagues have shown, and you can see that on the right-hand side of your screen, that liver-resident macrophages, known as Kupffer cells, they promote oncogenic transformation in a JNK-dependent manner and facilitate CCA proliferation and progression. There are a number of factors that are associated with augmenting abundance of TAMs, and these include certain chemokines and growth factors such as colony-stimulating factor 1 receptor, and we'll come back to this a little bit later in the talk. So this was a single-cell RNA sequencing analysis on 14 patients with intrahepatic cholangiocarcinoma, and it demonstrated that there are two distinct molecular subtypes of cholangiocarcinoma that are really defined by two markers, S100P and SPP1. And the subtype where there was expression of S100P was termed the perihilar large duct subtype, and this subtype correlated with poor patient outcomes. And there was an abundance of CCL18 positive macrophages in this subtype. On the other hand, the subtype that was defined by SPP1 expression was a perihilar small duct type, and it did not have an abundance of these types of macrophages. This was a multi-omic analysis using whole exome sequencing, proteomics, on 151 patients with intrahepatic cholangiocarcinoma. Some of these patients also had single-cell RNA sequencing conducted, and this multi-omic analysis identified three molecular subtypes: and inflammation, metabolism, and chromatin remodeling. The inflammation subtype was characterized by the presence of ApoE positive, C1q-B positive macrophages, and the patients that had these macrophages and had this subtype had worse outcomes. So we know that macrophages, particularly certain subsets of macrophages, are associated with patient outcomes, and specifically poor patient outcomes. The other myeloid cell that I'll focus on today is myeloid-derived suppressor cells, or MDSCs. And there are two primary subsets of MDSCs: granulocytic, also known as polymorphonuclear, or PMN MDSCs, and monocytic, or MMDSCs. And these are immature myeloid cells that foster tumor growth by suppressing cytotoxic cells, such as CD8 T cells, and promoting tumor cell proliferation. And there's data from other cancer types that suggest that high levels of circulating MDSCs promote resistance to immune checkpoint inhibition. And this was very recent work published by Bridget Keenan and the wonderful group at UCSF, including Katie Kelly, showing that a subtype of these monocytes, which express CD14, their abundance increased in patients who did not respond to immune checkpoint inhibition. So it was associated with resistance to immune checkpoint inhibition. And this work was, this population was identified primarily by using CiteSeq. So work done in our lab, we've shown that that macrophages, or TAMs, especially particularly those that express PD-L1, are essential for CCA tumor progression. And we demonstrated this in both murine models of cholangiocarcinoma, where you can see that in mice that did not have PD-L1, a total body knockout of PD-L1, there was a significant reduction in the tumor burden when we implanted murine CCA cells that express PD-L1, suggesting to us that PD-L1 positive host immune cells are really important in promoting tumor progression. And when we used human specimens and co-stained for macrophage markers and PD-L1, we found that the majority of PD-L1 positive expression occurs on macrophages rather than cancer cells. So TAMs are the predominant source of PD-L1 in CCA based on this work. And given that knowledge, we tried to block TAMs using both genetic means as well as pharmacologic means using anti-CSF1R. So that's the growth factor that I showed earlier. We tried to block it. And we did not observe any change in the tumor, which was a surprise to us. And this occurred because there was a compensatory emergence of GM-DSC. So this tells us that there's extensive crosstalk between these different immunosuppressive cell types. They're friends of the tumor. They connect with each other to promote tumor progression. So when we try to block one, another one comes up. And then using an unbiased screen, a chemokine screen, we found that in the tumors that were anti-CSF1R treated, there was a significant increase in the chemokine CXCL2, and then we followed that with fluorescence in situ hybridization or FISH analysis and showed that the primary source of CXCL2 in the mouse CCA tumors were alpha smooth muscle actin positive cancer-associated fibroblasts. So again, demonstrating the intricate crosstalk between all these different types of cells in these dense desmoplastic tumors. And so having demonstrated this compensatory emergence, then our next step was try to see if we could block both of these populations, both of these myeloid cell populations, and whether that would improve the effectiveness of immune checkpoint inhibition in our mouse models. And so we conducted several studies and used a variety of treatments blocking either the macrophages by themselves, the MDSCs by themselves, macrophages plus MDSCs, and then combining a variety of combinations with and without immune checkpoint inhibition. And it's when we blocked both macrophages and MDSCs that we noted a significant improvement in the response to immune checkpoint inhibition and prolonged mouse survival. And we confirmed that using micro-CT scans, which you can see on the right-hand side of your screen, or left hand. Is that left? Yeah. And so then we went on to try LX receptor agonism. So LXR agonism has been shown to promote apoptosis or cell death of MDSCs, and this was work that was done by Sohail Tavosi at Rockefeller, showing that LXR agonism using a pharmacologic inhibitor known as GW3965 promotes MDSC cell death, and there is an LXR agonist that's now in early phase clinical trials for solid organ malignancies. And so using this LXR agonist, we found that when we combined this with macrophage blockade, we had very similar results to what we had observed when we tried to block MDSCs using a neutralizing antibody known as anti-Ly6G, and you can see those results in that graph. And so to conclude, with this work, we showed that murine CCA tumors, as well as human tumors, have an abundance of PD-L1 positive TAMs. These TAMs promote dysfunction of CD8 T cells, and when we try to deplete these TAMs, we have a compensatory emergence of the other myeloid cell population that's really important, MDSCs, and then when we blocked both of these populations, we were able to improve response to immune checkpoint inhibition. And then this is a very nice paper that was published by David Linehan, and this was published in Gut in 2021. And similar to our findings, they found that TAMs are the predominant immunosuppressive cell type in cholangiocarcinoma, and they showed this both in mouse models as well as human specimens. And tumor-derived granulocyte macrophage colony stimulating factor, which is a growth factor, that expression of GM-CSF is prognostic in CCA. And neutralizing GM-CSF reduced murine tumor growth. There was these tumors that were treated with the GM-CSF had decrease in abundance of TAMs and also had repolarization of immunosuppressive TAMs and MDSCs. So again, highlighting the importance of these myeloid populations in promoting tumor growth. So this was a paper that was by Tim Gretton's group, and it showed the interplay between the gut microbiome, MDSCs, and their role in promoting cholangiocarcinoma progression. They showed that in preclinical models of both primary sclerosing cholangitis, or PSC, and colitis, there's an increase in hepatic CXCL1, that's a chemokine, and this occurs in a TLR4-dependent manner. And it accelerates CCA growth in these preclinical models. And this could be reversed by either antibiotic treatment to modulate the gut microbiome, neutralizing that chemokine, or hepatic TLR4 knockout. And they also demonstrated that high levels of CD15 positive myeloid cells in PSC patients with are present in PSC patients who have active colitis compared to those who have very quiescent or quiet disease. This was also work by Tim's group looking at CD40 agonism. So CD40 activates antigen-presenting cells, such as dendritic cells, repolarizes macrophages to a more of a anti-tumor type. And using CD40 agonism in mouse models, they demonstrated increased activation of both macrophages and dendritic cells, as well as increased activation of cytotoxic cell types, such as CD8 T cells. And they then went on to combine CD40 agonism with the standard of care therapy at that time, which was gemcitabine cisplatin, and combined that with the immune checkpoint inhibition, and found that the quadruple therapy resulted in even more robust tumor suppression. So this was, again, all work done in mouse models. So the rest of the time I'll spend on some of our unpublished work, again, focusing on myeloid-derived suppressor cells. So tumor necrosis factor-related apoptosis-inducing ligand or TRAIL, also known as a mouthful, is a member of the TNF family of proteins. And TRAIL receptors are expressed on immune cells, which use it as an important mechanism to induce cancer cell death in cancer cells that express the receptor. And so immune cells express the ligand, and TRAIL receptors are expressed by the cancer cells, and that's how immune cells try to kill the cancer cells. However, there has been extensive interest for over 2 decades on TRAIL agonism, and all TRAIL receptor agonists have universally failed in human clinical trials. And so we wanted to look into this a little bit further, because it led us to hypothesize that, that perhaps there's a dual role. TRAIL has both a pro-tumor role, as has the known anti-tumor role, which is cancer cell death, but perhaps there's a pro-tumor role in terms of modulating the tumor immune microenvironment. So using several different mouse models of cholangiocarcinoma, 2 of which are highlighted on this slide, we showed that when we implant mouse cells with different genetic backgrounds, so that's YAP-AKT, KRAS, P53, and then there's a third model that we have using dominant negative FBXW7, along with AKT, all three models showed the exact same thing, that when we implanted murine cells into these mice, there was a significant reduction in tumors in mice that had genetic knockout of trail receptor. And so that's really counterintuitive, right? Because trail receptor is known to induce cancer cell death. And here we are trying to, having knocked it out and showing that tumors are smaller. And so that went along with this hypothesis that maybe there is a dual role here, and that maybe trail receptor on the immune cells is essential in promoting CCA progression. And so this was really the hypothesis that maybe there's a divergent role. Yes, trail can induce cancer cell death, but there may be an effect on the immune microenvironment that is pro-tumor, and perhaps that's by fostering immunosuppressive cell types. And so indeed, when we profiled these tumors in the trail receptor knockout mice, we found that there was a significant reduction, again, of these MDSCs. So it just highlights how important these cell types are. And both the GMDSCs and MMDSCs were significantly reduced in mice that did not have trail receptor. And then we went on to specifically knock out the receptor using a conditional knockout mouse model, specifically knock it out in myeloid cells, so macrophages and MDSCs. And in these mice, and that's the lower graph, our results were very similar to mice that had total body knockout of trail receptor. And when we profiled these mice, they also had a decrease in both GMDSCs and MMDSCs. And then to demonstrate that this was not an intrinsic effect due to a total body knockout of trail receptor, and it's really related to the effect of trail deletion, we then took murine CCA cells and employed CRISPR-Cas-mediated knockout of trail as well as trail receptor, and then took those cells and put them back in the mouse. And we found that when cancer cell restricted deletion of trail resulted in a significant reduction in the tumor. So that's the middle bar on that graph. And on the other hand, and this is interesting and similar to what is known about trail receptor, right? When we knocked out the receptor, there was a significant increase in the tumors, and that's because that's the arm of trail signaling that is required to cause cancer cell death. And so with mice where the cancer cells didn't have the receptor, the immune cells could not kill the cancer cells. And so in summary with this work, and this is all unpublished work that we're currently in the process of completing, we've shown that there's a divergent role of trail-R in the tumor immune microenvironment of cholangiocarcinoma, whereby trail-positive cytotoxic immune cells can trigger cancer cell death. But on the other hand, trail-positive cancer cells can leverage trail signaling, and this we have data showing that this is primarily non-canonical trail signaling rather than the canonical cell death signaling to evade the anti-tumor immune response. And so what have we learned? And there's been a plethora of literature on the tumor immune microenvironment and a lot of very nice studies published specifically in cholangiocarcinoma looking at myeloid cells in the last, I would say, really 3 years. And so from these studies, we know that CCAs are very immunosuppressive. Tumors, there's an abundance of myeloid cell types. And a variety of preclinical studies have shown that there are perhaps some therapeutic strategies that we can leverage that may re-engineer this immunosuppressive tumor immune microenvironment to an immunogenic one that responds better to immune checkpoint inhibition. So with that, that's my last slide. I thank you very much for your time.
All right, thank you so much to all three speakers for a series of wonderful talks. And as the audience can see, targeting the immune system in cholangiocarcinoma is analogous to playing whack-a-mole. If you knock one thing down, it comes up. So I believe at this point, Stacy's going to ask us some questions, or I'll lead the panel discussion and sit back down and take some questions for the audience. And if no one has any burning questions, we have some things that we can talk about publicly and get input. Thank you.
Hello. Stacy is not leading the discussion. Thank you, Greg, Ben. Thank you, Ben. If you have questions, please go to the microphones or please raise your hand and we'll get a microphone to you.
One question. Great session. Thanks, everyone. So there's been a long-standing interest in blocking chemokines that recruit myeloid cells, CXCR2 inhibitors, CCL2 inhibitors. There's been talk for a while in many solid tumors about those, but I haven't heard much from the clinic lately, so I guess if anyone in the panel has insight into how those myeloid chemokine inhibitors are doing or not doing, be helpful to know.
V had a big smile on his face. Go ahead, V. Or Anna.
Do you know it? I'm certainly not aware of any myeloid trials going on, myeloid inhibition trials. Do you? No.
Sorry.
I mean, there have been trials using, anti-CSF1R with the hope of targeting myeloid cells, but what we've learned from those trials, even from other malignancies, that it's a therapy, one, that hasn't really been as effective, and second, it's really not very well tolerated.
I mean, I think Dave Linehan published on CCL2 inhibitors, if I'm not mistaken.
GM-CSF neutralization. And so I think Katie had that was GM-CSF plus Pembro, right? You had a study. Agnes, yeah. Okay, so it's the opposite. So.
And actually, I wanted to ask about that. I mean, I think this highlights the complexities and unanswered questions, right? We have data that GM-CSF is immunosuppressive. We have data that it supports effector cell function. Which direction should we go? And, I think Katie had a 21% response rate with GM-CSF plus nivolumab, or plus Pembro, which is higher than you would expect with PD-1 monotherapy. So do we really understand, or do we need to go back to the lab? I think it's a good question. Katie, I don't know if you have any comments.
A very small study, but also history as well. And so I think that I don't have an answer at all other than it may be milieu context dependent. One small finding from our study is responders were more likely to have viral hepatitis in our cohort, which may be a different issue related to the PD-1 response, though that jury is still out too.
Yeah, and that, again, it highlights the complexity, and our group has data showing that certain cholangiocarcinoma cell lines from humans, when you put them in culture, they'll spit out nanogram quantities of GM-CSF. So in certain patients, it may be very high in the local tumor microenvironment, while in others, it may be lower. And this variability, both in the genomic state of cholangio, the effect it has on the immune profile of the tumors, all these things may matter, and I think it really is pointing to a personalized approach, not just for targeted therapies, but potentially for immunotherapy. I think there's a question there.
And if I may, we still don't know if the immunosuppressive phenotype that we see is the egg of the chicken. We don't know if it's an effect still, or who is really determining that immunosuppressive environment, and if is the T regulatory cell, is the myeloid cells, are the fibroids, the fibroblasts influencing the microenvironment. So still it's a very complex situation in which we cannot think that one player is running the whole show.
Yeah, and the only other thing to add from the bench side, I think it also, going back to the point you made about a personalized medicine approach, I think it emphasizes that we do need more than, one mouse model. We need different models, have better preclinical models, continue to develop these and have models with different genetic drivers, and then try to see which one of those can correlate to a subset of human cholangiocarcinomas.
I am constantly disappointed that my favorite protein isn't showing up among the ones listed. My favorite protein is PIN1. I like it because when it is inhibited in mice with pancreatic cancer, the CAFs pretty much disappear. Macrophages change from M2 to M1. Tregs are very much reduced, and when you give that along with chemo, and especially with immune therapy, anti-PD-1, 8 out of 9 of the mice have complete remission, and they live for a whole year, even when you stop the therapy after 4 months. We know, there is evidence with a explained mechanism that in cholangiocarcinoma with active KRAS, you have overexpression of PIN1. And there is this evidence in pancreatic cancer that when you have overexpression of PIN1, the tumor microenvironment is immune suppressive. And if you inhibit this expression, it becomes immune supportive. Should we have some study of whether this kind of thing might also be helping with cholangio?
So I think it kind of goes back to that point, right? Cholangiocarcinoma is not one disease or one tumor type. As you mentioned that your favorite protein is overexpressed in the setting of KRAS mutations. And so we know that only a subset of cholangiocarcinomas are KRAS mutated. And I think that, which kind of goes back to that point of trying to have different models in the preclinical setting that have different genetic drivers and try to dissect those, because there may be another mouse model that we look at it. You know, you mentioned one study in pancreatic cancer where 9 out of 10 mice responded. Well, I can tell you there's tons of studies out there where, you know, in a mouse model, blockade of a protein or an immunosuppressive molecule led to a reduction in the tumor burden, but the majority of that doesn't correlate to patient outcomes. And so I don't know if, you know, this is really the protein that would, I think it could be important in a subset, but I think it's hard to say whether this would translate to the majority of cholangiocarcinomas.
Let's get to the burning question in the front here.
I have a quick question on the upregulation of Treg phenotype in intrahepatic cholangiocarcinoma. Do we know anything about the mutational landscape? Is this the case for IDH1 mutated iCCA?
Sorry, can you repeat the question? Do you know anything about?
Do we know anything about the mutational landscape of these patients? Any information if this would be the case in IDH1 mutated cholangiocarcinoma?
Yeah, that's a very good question. So all the patients that we included in our study were wild type, so none of them had mutations. I can imagine that there will be differences in the different subtypes, so ideally we should repeat the study for all the targeted mutations that we know, or probably, so we are currently expanding our data and including whole exome sequencing also to try to identify if one phenotype is related to a specific mutation. Thank you.
Thank you, Vincenzo Cardinale from Rome. Congratulations for this very beautiful session. I have 2 possible questions. One is from Professor Lleo, and do you think it's possible to target this Treg population with drugs that can complement actual treatment? This is the first question. Second question for the panelists. In ACC, there is a lot of interest of combination VEGF inhibition and immune therapy. I know there is something also in cholangiocarcinoma going on. I want to know if there is interesting news also in the combination of VEGF and immune therapy.
So thank you for the question. Well, the easy answer will be yes, we hope so, but I think reality is much more complex. So at the moment, we are studying what are the mechanisms that drive that immunosuppressive pattern in order to see if it's actually the beginning or the end of the process. We hope so. We also want to know exactly what is the antigen stimulation that is driven that stimulation or that immunosuppressive pattern. So there are still a lot of pathways that need to be identified in order to say we can specifically target that one. Because she could tell me, okay, what about my myeloid cells? So let's target those, and probably we will have an answer. So still, there's a lot of work to be done.
I'll take the second part of the question, which was, can we do combination anti-PD-1/L1 plus VEGF in biliary cancer and take that to clinical trials? So I believe at ASCO GI earlier this year, the trial results from gem-cis atezolizumab versus gem-cis atezolizumab plus bevacizumab were announced. There were no significant differences in survival or response rates. Thank you.
We have a question far in the back here.
Yes, so this is Li Qing from St. Jude Children's Research Hospital. So I have a question about the general idea of macrophage being immune suppressive. So, because in the immunity, we know that they are supposed to be our defender, right? So they should heal the wound. But in liver cancer, in most cases, they are actually become, not only they are not targeting tumor cells, they actually become protectors. So I'm wondering whether this has something to do with the liver itself, because it is known as an immune tolerant organ, so it tends to reduce the immune activation because of the constant insults that liver receives on a daily basis, and also because the fact that it has the largest resident macrophage population by nature. So I'm just wondering whether liver cancer is unique compared to other solid tumors in terms of its immunity actually kind of conveys the fact that somehow immunotherapy doesn't really work in this type of tumor.
Yeah, I think that's a great question. That's actually when I started out to set up my program several years ago now. That was one of the main things and that's actually the primary title of my main grant, which is the immune tolerogenic environment of the liver. And when we started out the work with the myeloid cells and specifically the macrophages, the overarching hypothesis at that time is that the liver is a very sort of at baseline, it's an immune, it's a permissive organ and it's immunosuppressive. And so our hypothesis at that time was that the resident Kupffer cells are a key in promoting cholangiocarcinoma, not only formation, but progression. But all the work that we did with the mouse model, and this is our published work now, we showed that the majority of macrophages in cholangiocarcinoma in mouse models and in the stainings that we did in the human specimens are actually recruited from the circulation rather than the resident Kupffer cells.
I believe Dr. Dretton had a question.
Yeah, actually, I just wanted to make a comment to the question about the VEGF combination, and I will talk about this tomorrow. And we have a study where we test this, and I think it's actually going to be very interesting, both for the scientists as well as for the patients.
So just in the last minute, I'll just ask my co-panelists here. There's so many checkpoint inhibitor molecules coming. TIGIT, LAG3, et cetera, TIM3. Do you think we've hit a ceiling with checkpoint inhibitors? I mean, obviously PD-1, PD-L1 is going to be the common denominator, but have we hit a ceiling with just broad checkpoint inhibitors, or is there more to come?
And let me add to that before you answer. I think as we move forward with TOPAZ and other regimens that are potentially coming down the line very soon, the reality is going to be in subsequent lines of therapy, the patients who will receive experimental immune therapy drugs or alternative checkpoints, if you will, they're going to come in as PD-1, PD-L1 targeted refractory patients. So that really ups the game of what we need to do from an immunologic standpoint. So curious to hear what people think.
I guess I'll start because I'm the first here. So my naive way of looking at this is we've come a long way in genomically typing our patients. I think we need to do immune microenvironment typing to answer what you're asking. There are patients that we've identified that have high TIGIT expression, but that's only a subset of patients. And if I give anti-TIGIT antibodies to all the patients, that will be a negative trial. So again, we need to identify which patients will benefit, in my view, which is why we're seeing discussions about anatomic subtypes. We're seeing discussions about genomic data helping us identify which patients would respond to immunotherapy. I think we need to take a step back and look at the immune microenvironments to answer these questions and divide that patient population into answering these questions better.
And the only thing I would add to that, I don't think that there will be one immune checkpoint inhibitor or one therapy that will work even for a subset of patients. I think the key will be in combinations, because we've seen that we know from preclinical and translational work that there's a lot of compensatory phenomena that occur. And so I think the reality will be having different combinations for different subsets of patients, not only molecular subsets, but also looking at the different anatomic subtypes of cholangiocarcinoma. I just don't think that there will be a one-size-fits-all solution at all here.
I have nothing to add to that.
All right, well, I think that sums up our session here. It's really an exciting time for immunotherapy, and I just want to thank all of my panelists here for great talks. Thank you.
Right, so now it is time for us to hear from three more of our fellows. The first I will introduce is Dr. Cecilia Monge. Dr. Monge is involved in clinical research focused on novel first-in-human and early-phase clinical trials within the realm of GI tumors and immunology. She has served as chair and investigator of multiple early-stage clinical protocols within the NCI and NIH, and she is going to be speaking to us about a phase 2 trial of Pembrolizumab and CDX-1140.
Hi, good afternoon, everyone. First of all, I would like to thank the Cholangiocarcinoma Foundation for the grant that I received a year ago. And from that grant, this protocol that I will present today has taken place. So, as has been discussed this morning, these are the standard lines of treatment for biliary tract cancer, both in the first line and the second line setting. And as has been mentioned, the second line varies according to if the patient has targetable genetic mutations or not. Our group has been studying immunotherapy in liver cancer, both in mouse models as well as in clinical protocols with humans. I'm going to show one of the immunotherapy studies using anti-PD-1 and combination with chemotherapy. So this was a phase 2 study that was completed by our branch, and we treated patients with advanced biliary tract cancer in the second-line setting, and they were treated with pembrolizumab as well as capecitabine and oxaliplatin. A total of 11 patients enrolled, and the median follow-up was 34.8 months. The study design consisted of 3-week cycles, and the patients received oxaliplatin on day 1 of each cycle, as well as capecitabine. And capecitabine was given for the first 2 weeks of each cycle, twice a day. Pembrolizumab was given on day 1 of each cycle as well with oxaliplatin. It's important to mention that the capecitabine and oxaliplatin was given only for 6 cycles of the trial, and then the patients continued on pembrolizumab alone every 3 weeks until progression of disease or toxicity. So we had 3 patients with a partial response, and the best partial response was 80% shrinkage of the tumor, and 6 patients had stable disease. The disease control rate was approximately 82%. The median progression-free survival in the study was 4.1 months, with a 12-month progression-free survival of 9.1%. And the median overall survival was 9.9 months, with a 6-month progression-free survival, with a 6-month overall survival of 81.8%. Grade 3 and 4 treatment-related AEs. The most important ones were lymphocytopenia, which we saw in almost 2/3 of the patients, anemia in 36%, and thrombocytopenia. So why are the results of this small clinical study important? Well, because they underline the importance of improving the efficacy of immunotherapy in biliary tract cancer. So we decided to study the use of anti-CD40 agonist antibodies to improve the efficacy of immunotherapy in biliary tract cancer. And we specifically looked at CDX-1140, which is a fully human IgG2 agonistic anti-CD40 antibody. And it's been developed by Celldex Therapeutics. So CD40 is basically a tumor necrosis factor receptor, and agonistic CD40 antibodies have the potential to generate anti-cancer immunity through various mechanisms. Basically, the interaction of CD40 with a CD40 ligand results in the activation of CD8 T cells and antigen-presenting cell activation. So this study from the Gretton lab was published last year by Dr. Diggs, which is actually also a Cholangiocarcinoma Foundation fellow, and he did, in the Gretton lab, the preclinical work for the protocol that I'm presenting today. And basically what was done was to hypothesize that combining a CD40 agonist with an anti-PD-1 could overcome the immunosuppressive microenvironment of biliary tract cancers. So to make the study as relevant as possible, 3 different models were used of biliary tract cancer. So one was subcutaneous, the other one was orthotopic, and the second one was induced by 2 plasmids. And basically what we can see in green is the effect of overall survival with anti-PD-1 alone, in blue with anti-CD40, and in red with a combination of anti-PD-1 and anti-CD40 in mice. And we can see that even though these mice were treated for a short period of time, the overall survival, especially in the mice that received the combination of anti-PD-1 and anti-CD40, is actually long-lasting. If we look at the tumor-liver ratios, we can also see that the combination of anti-PD-1 and anti-CD40 was the one that had the best response. So in humans, we give immunotherapy with chemotherapy. So these graphs show the efficacy of anti-CD40 plus anti-PD-1, and then when immunotherapy is added. And we can see that the tumor-liver ratios greatly decrease with a combination of all agents. So many times when we're studying tumors in mice, if the tumors are small, it's easier to see more effectiveness. So it's important to point out that in this preclinical study, mice were treated when the tumors were quite large. So they were almost 10% of the size of the mouse when the treatment started. And we can see that even though, again, the treatment period is short, if we look at the overall survival of the mouse when we combine GemCis, anti-CD40, and anti-PD-1, the overall survival is extended. And we see the same as far as the tumor-liver ratio. And this is the combination of the 4 agents is what we see in red in these 2 graphs. So we decided to analyze, from an immune cell perspective, what was causing this decrease in tumor growth. And for this, we did flow cytometry analysis. And what we saw was that with a combination of anti-CD40 and anti-PD-1, there was an increase in effector cells, specifically those producing interferon and those expressing CD107 as a degranulation marker. So as I decided to take this to the clinic as a clinical protocol, it was important to look at what was the experience of this specific anti-CD40 in humans. So there's an ongoing phase 1 dose escalation trial that combines anti-CD40 CDX-1140, which is the one that will be used in this protocol, with pembrolizumab. And the most important part is the toxicity part, which is the data that has been published. And basically, this is the most important part because with CD40, as we all know, there's always been a concern with toxicity in human trials, specifically with hepatotoxicity. So we've seen that in 20 patients that have been treated with this combination so far, 36% have had grade 3 treatment-related adverse events, and the most frequent of these has been transaminase increase, but it has been grade 3. 2 patients had grade 3 pneumonitis that resolved with steroids, and 2 patients had grade 4 treatment-related adverse events, which consisted of encephalopathy and hypoxia that was not associated with pneumonitis per the investigators. So basically, with the hypothesis that with CDX-1140, we can achieve a better priming of the CD8 T cells and the antigen-presenting cells, and therefore increase the effectiveness of immunotherapy and further increase the effectiveness of chemotherapy, this study came to take place. So the inclusion criteria basically are that patient must have received at least 1 line of treatment in the advanced setting, and they must have at least 1 or more measurable lesions, and this is for paired biopsies. Exclusion criteria include prior treatment with immune checkpoint inhibition, and this may be amended, and a prior CD40 agonist treatment. So the study design currently includes cycles of 3 weeks, and on day 1 of each cycle, patients receive oxaliplatin, capecitabine similar to the first study I showed you from our group, is given from day 1 to day 14 orally, and then patients will receive CDX-1140 with pembrolizumab anytime between day 7 and day 10 in the study. Importantly, the chemotherapy will be given only for 18 weeks, and then pembrolizumab and CDX-1140 will continue until disease progression or toxicity. The patients will undergo a pre-treatment biopsy and then an on-treatment biopsy in the last week of cycle 2, and we will image them every 3 cycles or 9 weeks. Our primary endpoint is to determine the progression-free survival of this combination therapy. Thank you.
So the next fellow will be Alessandro Gambella, who is an MD pathologist and PhD student at the University of Turin and a research fellow at the Division Liver and Transplantation Pathology at the University of Pittsburgh Medical Center. And he will give a talk with the title, The Role of Precision Pathology in Cholangiocarcinoma, the Role of Immunosynapses and Vascular Framework in Patient Management. Thank you.
Yeah, good afternoon, everybody. Thanks a lot for the kind introduction and to the CCF Foundation for having me today. It's really an honor and a pleasure. So here's a brief outline of my talk. We had a slight different approach. We started with non-neoplastic disease involved in the bile ducts, and then I'm going to show you why this is relevant and how it led us to study the immune synapses and the vascular density. So my mentor and I are pathologists, and as pathologists, we look at the tissue and the samples to look for cell interaction. And if we look at cell interaction of the immune system with the bile ducts in non-neoplastic disease, there's actually a lot of evidence that show how the immune system can efficiently and completely destroy the bile ducts. And there are 2 major mechanisms behind that. One is a direct injury of the lymphocyte to the bile ducts. And you can see here in the top left image a normal bile duct that underwent an acute active injury by lymphocytes in the top left image that eventually lead in chronic inflammation to the complete destroy of the bile ducts, as you can see in the top right image. The second mechanism is that of the destruction of the peribiliary vascular plexus, which are the vessels that nourish the bile ducts. So whenever they are gone, the bile ducts suffer and they're injured. And you can see here in the bottom right graph that in the normal liver, the density is normal. And as much as the inflammation became chronic, the vascular density decreased. So as cholangiocarcinoma is a malignancy that originates from the bile ducts, from the cholangiocytes, our research question were whether these mechanisms are preserved or altered in the cholangiocarcinoma, and if we can boost them so that we can achieve this complete destruction of tumoral cholangiocytes as well. To answer this question, we collect about 100 cases of surgical resected cholangiocarcinoma, and we studied them from a genetical perspective, so with the mutation of the genes, with more traditional and advanced histopathology technique, and also with the transcriptome, the RNA, so how the genes are translated. The first data that I'm going to show you is related to MHC2. MHC2 is a molecule that when it's expressed in the bile ducts, it makes them visible to the lymphocyte so that the lymphocyte can find the bile ducts and destroy them. So we expected to see that patients where the tumor cells were positive to MHC2, they should have an improved survival. And here in the top left image, there is a tumor that is negative to MHC2 and is blue. And in the bottom left, a tumor that is positive, so it's brown. And this is an immunohistochemistry, so we evaluate the expression of the protein in the tumor cells. But when we look at the effect of this expression of patient survival, we found something unexpected. So that patients that had cholangiocarcinoma tumor cells positive to MHC2 actually had a worse survival. So we analyze deeper the reason behind that paradoxical outcome. And what we observed is that the immune environment of the cholangiocarcinoma with MHC2 positive tumor cells were characterized by inhibited lymphocytes, specifically lymphocytes that express PD-1 and LAG-3. So from our perspective, we hypothesize that this is quite relevant because we believe that this subgroup of patients with MHC2 positive tumor cells would mostly benefit from immune checkpoint inhibitors, so that when we remove the block from PD-1 and LAG-3, the lymphocyte can target visible tumor cells. Another molecule that we were interested in is galectin-9. Galectin-9 is an immune modulator. It interacts with TIM3, and by interacting with TIM3, it inhibits the immune response. So again, here we expected to see that whenever galectin-9 was high, the survival was low. Still, we evaluated with immunohistochemistry. You can see again in the left, negative tumor cells blue and positive with different shades of brown in brown. And on the right side, you can see the effect of galectin-9 expression on the survival of patient. And again, we saw a paradoxical outcome, so that whenever galectin-9 was high, the survival actually was improved. So again, we look at the potential mechanism behind that, and we saw that there was an inverse correlation between galectin-9 and TIDO2. So here, the two graphs are going to show you the same message. And the message is that whenever galectin-9 is high, TIDO2 is low, and vice versa. On the left side, you can see the transcriptomic analysis. On the right side, the immunohistochemistry, so the protein expression. This is relevant because TIDO2, together with IDO, are enzymes that regulate the level of tryptophan. And tryptophan is a molecule that is essential for lymphocytes to grow and to perform their action effectively. So this allows us first to justify the results that we saw, so that whenever the TIDO is low and the immune system actually have tryptophan and can efficiently destroy the tumor cells, galectin-9 is low. And on the other side, when TIDO levels are low, the tumor cells start expressing galectin-9 as an immune escape mechanism. The other relevant findings based on this data is that actually there are target therapies that can target tryptophan catabolism. So we believe that it would be actually interesting to evaluate even further this mechanism in cholangiocarcinoma. So now that we have evidence that the interaction of lymphocyte and tumor cells is actually essential, we decided to evaluate immune synapses. Immune synapses are actually the most sophisticated and specific tools that we have to evaluate the activation of lymphocyte. There are almost as the synapses in the brain, so what they do is actually, it's a way to identify a communication between two cells. In the immune synapses, the cells that are communicated are a lymphocyte and a target cells. And specifically in cancer immunology, the target cell is, of course, the tumor cell. So we have a cytotoxic synapses whenever the lymphocyte recognize the tumor cells and release granzyme B to destroy the tumor cells. But unfortunately, the tumor cells are able to evade this mechanism so that they can induce the expression and co-localization of inhibitory molecules where the granzyme should be released, so that these granzymes are not released and the tumor cells are not destroyed. So to evaluate the immune synapses is actually quite complex, and we need to develop a next-generation pathology tool that allows us to evaluate multiple markers on the same cells. Here on the left part of the image, you can see in the top row a cartoon that shows the cytotoxin synapse and the exhaustion synapse. And in the lower row, you can see the corresponding image that we saw directly on tissue. While on the right side, there is the digital pathology algorithm that we developed to identify those synapses and to correlate their localization within the context of the tumor cells. And here is also where the vascular density will be integrated, because as I told you before, immune synapses are between a lymphocyte and a target cell. And the target cell, in this case, could be also represented by the vessels. So in conclusion, we identified that it could be interesting to further evaluate this mechanism of bile duct destruction and how they are different in the cholangiocarcinoma immune environment, specifically the role of MHC2 in this case, and how we could identify a niche of patients responsive to immune checkpoint inhibitors, and the relation between galectin-9 and tryptophan. Regarding immune synapses, so far we have identified them in the tissue, and this is quite relevant because there are very few data, if none, about the identification of immune synapses directly on tissue samples. And we are still developing the further characterization and their prognostic and predictive role. With that, I conclude. I just want to thank you again, the CCF Foundation, because it really has a huge impact not only on this research project, on my personal path as well. And I also would like to thank my mentor, Dr. Dimitris, and all the amazing colleagues and people that work at the Liver and Transplant Pathology Unit in Pittsburgh, that they constantly support me daily in this research project. Thanks.
All right, our final fellow presentation for the afternoon is from Marina Barcena-Varela. Dr. Barcena-Varela is a postdoctoral fellow at the Icahn School of Medicine at Mount Sinai in New York. She's an active member of the Young Investigator Steering Committee from the International Liver Cancer Association. She's going to be speaking on harnessing the microbiome for precision cholangiocarcinoma therapy.
Okay. Good afternoon, everyone. Thank you very much for the introduction. And I'm very happy to be here today presenting this project that is focusing on harnessing the microbiome for precision cholangiocarcinoma immunotherapy. As very brief background, because you are all familiar with this, cholangiocarcinoma is the second most common primary hepatic malignancy affecting the bile ducts of the liver, with an increasing incidence globally. There is variety of risk factors for CCA, which make this disease particularly heterogeneous. And its silent presentation and highly aggressive nature make it very complicated to diagnose at early stages, so patients are generally diagnosed at the late time points of the disease, where only about 25% of patients are eligible for surgical resection. So with all this background, I just want to highlight that there is an urgent need to discover novel therapeutic approaches for CCA patients. Cholangiocarcinoma can be divided by the anatomical site of origin that you all know. Intrahepatic cholangiocarcinoma is the most common subtype. Extrahepatic cholangiocarcinoma include perihelial and distal CCAs, but I want to also mention here that there is this rare mixed HCC-CCA phenotype that include both features of intrahepatic cholangiocarcinoma and hepatocellular carcinoma. In addition to that, CCAs are very heterogeneous within each of these subgroups, and I want to highlight here the work published by Daniela Sia that has previously highlighted in other talks, where she developed a novel microenvironment-based subclassification of intrahepatic cholangiocarcinoma that involves aspects of the microenvironment, the stroma, the tumor, and the immune cells. And as it has been discussed in the previous panel, the microenvironment is critical and very relevant for the response to different therapies, and especially for the novel use of immunotherapy for cancer patients. So immunotherapy is a revolution in the management of many cancers. However, as it has been mentioned before, the response rates for cholangiocarcinoma patients remains dramatically low. And in this context, it's very important to understand the molecular mechanisms of the microenvironment of these tumors that might be participating to the different response of these cancers. So here we have a general separation of hot tumors and cold tumors, being the hot tumors, the immune-like phenotypes most likely to respond to immunotherapy. Here there is a table that I'm not going to go in deep with it, but it's just summarizing the completed and ongoing clinical trials for immune checkpoint inhibitors in cholangiocarcinoma patients. And what I want to mention here is that now there are more attempts to combine immune checkpoints inhibitors with different approaches, and combining them also with chemotherapy or targeted therapies, and it has been just discussed in the previous panel that this combination therapies is probably going to be the best approach for cholangiocarcinoma patients, especially because of their diversity. So in this context, our work is focused in understanding what role gut microbiome can play in all these different responses to the immunotherapy, because gut microbiome is emerging as a potential modulator of the anti-tumor immunity and the response to immunotherapy in many different cancers, including liver cancer. It has been demonstrated that different microbiomes can affect significantly response to anti-PD-1, but it's been studied in other different immune checkpoints inhibitors. And in the context of the liver, and especially in cholangiocarcinoma, I wanted to highlight here in these publications that it has been observed that the microbiome is able to modulate the recruitment of immune cells in the tumor microenvironment and the activation of these immune cells in the context of preclinical cholangiocarcinoma models. So our hypothesis is that changes in microbiota composition affects cholangiocarcinoma tumorigenesis, progression, and response to therapy, and that these effects might be exploited to improve response to immunotherapy. To prove this hypothesis, we are following two main aims. The first one is to study how microbiome affects the cholangiocarcinoma tumorigenesis process, depending on the genetic alterations. For that, we are using precision mouse models of cholangiocarcinoma, and then modifying the microbiome by treating the animals with antibiotics or colonizing them with fecal stools or specific bacteria populations. The second aim of this project involves the study of how this microbiome alteration can affect the response to targeted therapies, especially focusing immunotherapies using combinatorial therapeutic approaches in vivo. So there are several mouse models designed to preclinical studies of cholangiocarcinoma. However, there is additional efforts required to develop more relevant clinical models for CCA that can recapitulate the diversity of the genetic alterations and the diversity of the phenotypes observed in CCA patients. So in this context, our laboratory, Lujanbio's lab laboratory, we develop precision genetically engineered mouse models to develop liver cancer, including both HCC and CCA. And the technique, how we generate these precision genetically engineered models, is by hydrodynamic tailbane injection of plasmids that can be directed to overexpress oncogenes or to silence tumor suppressor genes that are observed in patients. So in this project, I'm going to present data from 3 different models that we have initially used, but we are planning on testing in other different models that we have in the lab and we are currently developing. But here we have 2 well-defined and widely used models of cholangiocarcinoma that are based in the overexpression by SV13 transposon vectors of NICD and AKT, or the overexpression of JAP-AKT. And here we included also a third model that we have generated in the lab, and it's a very interesting model because it resembles the mixed HCC-CCA phenotype observed in patients. So very briefly, this model that we generated is based in the overexpression of MYC and NICD. And when we look at the transcriptomic level of a cohort of patients that are diagnosed as HCC-CCA phenotype compared with HCC patients, we observed that these patients have an overexpression of the Notch signaling pathway. They have also overexpression of cholangio-like genes, whereas downregulation of hepato-like phenotype genes. And we have performed similar transcriptomic analysis in this MYC-Notch model compared with HCC models, and we have confirmed that they resemble this phenotype. I'm not going to go in deep explaining the characterization of this model. Here, this amazing PhD candidate that we have in the lab, Catherine Lindblad, has worked in the development and characterization of this model, and this work is about to be published, hopefully anytime soon. It's under revision, and I encourage you all to read it whenever it came out, because I think it's going to be very interesting for future studies. So moving forward to the microbiome, so we have these models, and we wanted to study how these mouse models of cholangiocarcinoma can respond to different antibiotic treatments or colonization with different bacteria populations. Today, I'm only going to present some data of the antibiotic treatments because of the lack of time and also because the optimizations are taking long, but I will be happy to discuss. If anyone is interested about this offline, you can reach me in the conference and we can discuss about it. So moving to the results. Oh, sorry. Yeah. Moving to the results, the first thing we do was a survival experiment with mice that were injected with the indicated plasmids and treating them ad libitum with 2 antibiotics that we selected previously, neomycin and vancomycin, that we previously observed that don't have a major toxicity or side effects on chronic administration. And in the context of the AKT NICD overexpression, we didn't observe any significant difference in tumor development or survival on the mice. But very interestingly, when we moved to test the same antibiotics in mice injected with AKT and JAK, we did observe a reduce in median survival of the treated mice compared to the control mice. So mice started developing tumors earlier, and they die faster than the control group. But then, when we tested the same antibiotics in our novel model of MYC and NICD overexpression, we surprisingly observed the opposite effect. So in this case, neomycin and vancomycin treatment increased the median survival of MYC notch-derived tumors. So the mice started developing the tumors later and die slowly compared to the control group. So taking together the summary of the antibiotics effects we observed, we did not observe any significant changes in the tumor development in a context of AKT NICD tumors, whereas we did observe a pro-tumoral effect in an AKT and JAK tumors, and an anti-tumoral effect in MYC NICD-derived tumors. So the conclusions we have from this data that I saw here today, we have developed a novel mouse model of primary liver cancer triggered by the overexpression of oncogenes MYC and NICD in a hepatocyte that resembles the intrahepatic cholangiocarcinoma HCC mixed phenotype. We have demonstrated that altering microbiome composition by the use of antibiotics can significantly affect the tumor development in mouse models of cholangiocarcinoma. And finally, and very interesting, these effects to microbiome manipulation are highly determined by the genetic background of the cholangiocarcinoma tumors and can be either pro-tumorigenic or anti-tumorigenic. So we are already working in the next step of this project. So we are identifying the bacteria populations responsible for the observed phenotype. And as I said, we are colonizing the mice with specific bacteria. And we are obviously selecting bacteria that are observed also in patients, so we can translate our findings to the patients. We are also very interested in address how this manipulation of the microbiome can synergize with immune-based therapies, and to do that, we are going to combine this microbiome manipulation with different immune checkpoints inhibitors. And finally, we are intrigued to understand how genetics play a role in the different sensitivities to microbiome changes, because we really believe that understanding better why the effects can be dramatically different based on the genetics or phenotypes of the tumors may really provide information for better approach for the patients in a more personalized, oriented manner. So I want to thank everyone that has collaborated in this project, especially to Amaya Lujambio for the mentorship, and I want to highlight the work that Katie Lindblad have performed in the lab with the generation of the HCC-CCA model, and of course, thank you for the Cholangiocarcinoma Foundation because of the financial support for this project, and also for giving me the opportunity to be here enjoying this amazing conference, and also for presenting the data and giving me opportunity of networking. So thank you very much, and we're happy to discuss.